ATP turnover. We measure everything. So there's 750 kinases to measure and about 10 phosphatases to measure. So we can tell if the kinases are on or off. We can tell if the phosphatases are on or off. How do we do that? Wouldn't it be terribly difficult? Isn't that a horrible trial? Don't you at least have to think about this? No! What do? You were involved in the omics really well the first time. You do omics, you measure them all. You just measure them all. If they're on, they're phosphorylated. If they're off, they're not phosphorylated. I could pick out of the 750 unless I could measure them all. So we measure them all. The processes that turn off we already know because physiologists have already done the hard work. Holy Hannah! Gene inactivation. How will we discover that? I'm going to go to the message RNA and find what's turned off. This couldn't be easier, but I'm like Penn and Teller. Penn and Teller do magic, and they will show you how the magic trick works. You have no chance of doing that trick. So this is the trick. Anybody can do it, but you're not willing to. It's so easy. Protein kinases come in two families. One of them are called second messenger protein kinases, cyclic AMP, cyclic GMP, calcium IP3. And the other one are daisy-chained kinases. They phosphorylate. So this is to show you how mindless biochemists are. There's a protein called MAP. What is the protein that phosphorylates MAP called? MAP kinase. MAP kinase. What's the protein that phosphorylates MAP kinase? MAP kinase. Come on! Come on! What's the protein that phosphorylates MAP kinase? Yes, you're right. And there's a kit to measure them all. Just measure them all. You don't have to worry about which stress-activated kinases is on. You just do it. It just comes out in a big mask. Okay, here's a quiz then. Here's part of the quiz. We have to measure all of the activities of the protein kinases that are second messengers. True or false? False! We just take every tissue we have, under every condition we have students that are starting out homogenize everything, and we measure cyclic AMP in a kit. Ta-da! If cyclic AMP goes up, it's on. If not, it's off. Then we measure cyclic GMP in another kit. Then we measure, okay. So this is easy to do. This can be done. If it wasn't for a fear of molecular, everybody would do it. I think it's a fear of molecular. It's so easy. These are the protein kinases that actually turn on in every animal. It's the most amazing thing. These do different tasks. Do any of you take metformin? Metformin is a drug that regulates your carbohydrate versus fat use. If you're a diabetic, they turn on MAP kinase like they were diabetic. The mTOR pathway is the pathway of insulin. The core pathway to turn every animal off is the insulin pathway, and its tiny little friend, AKT. Jun and ERK turn off genes, and P38 is a stress kinase pathway. So you do all these animals, and you would think it would be terrible because you find the same thing again and again, but it isn't. You can publish new papers, and it's sort of straightforward. Well, I know you've measured these things, but they can't be important in every pathway. Oh yeah, every pathway. Every pathway you know is regulated by this. Pick up a biochemistry textbook and open it at any chapter. Every pathway is regulated by these protein kinases. In our animals, glycolysis is largely turned off. You change the phosphorylation status of many enzymes in the pathway to turn them off. Glycogen phosphorylase is off. Glycogen synthetase is off. PFK1 is off. PFK2 is off. Aldolase is off. DAPTH is off. Pyruvate kinase is off. What about the mitochondria? You can't shut down a mitochondria. Yeah, you can. You just turn off pyruvate dehydrogenase. Every pathway you know of, including transcription and translation, including ion pumps, are shut off by this very simple mechanism. What we find when we take human cells in a dish, and we haven't got enough human babies yet, it's hard to get your N up. once the parents know what you're doing with them. You turn on too many protein kinases. Your cells panic when your cells feel ice coming on them and they start to shrink down. When your cells get below a certain temperature, they panic and turn on all the protein kinases. The cool thing about our animals is these are the ones we turn on and we don't bother with the others. Also, you have to turn on all your genes. That was the other expensive thing. Half this audience has one organ that right now is actually doing what I'm going to tell you. It's one organ. It's taking perfectly good cells, turning off the nucleus. Turning off the nucleus, making it small. Tying the genes so they're not expressed, and pushing those cells out for export. One half of the audience, one organ or tissue. Talk among yourselves. Do you know? It's the male half, and it's what males are thinking about right now. Yes! You've got it right! Every single cell in this animal, including their neurons, turn into sperm. Sperm start out perfectly good. What happens is they bind up the DNA of their nucleus. They turn everything off, and then they send it out, hoping for the best. This process is called epigenetics. I worked on this for years, and I didn't know it was epigenetics, so I'm not the smartest guy. When you're an awake ground squirrel and an asleep ground squirrel, you have the same DNA sequence. You're not allowed to change your sequence and then come back for more. That sequence, though, can be physically modified. It can be methylated by itself. Did you used to smoke when you were young and foolish? Your DNA was methylated. If you gave up smoking 40 years ago, your DNA is already still methylated. Unless you're very lucky, it's going to be passed on. What frightens your grandmother actually shows up in grandchildren because DNA has been modified. These modifications are transgenerational. In our animal, they're not. They turn on and off. You put a big, bulky group called a methyl group onto DNA. DNA used to bind transcription factors. Now it just has a big, ugly block. You can't turn on genes. They don't turn on. You take histones, which are proteins that bind up DNA and cause it to form into what are called nucleosomes, and you just put them on steroids. You change histones, and you can phosphorylate them so that genes don't turn on. And you turn on microRNAs. MicroRNAs are like Zika mosquitoes. They arise in a cell as the cell turns off, and they destroy messages. They cover up transcription and translation. They just turn everything else off. That's what you do, and you do it in all cells, and it's reversible. So, it starts with that idea about energy expense. If you're going to take a cell and you have a problem with tens of thousands of species that have to stop making ATP and using ATP, this is a perfect method to do it, and all you have to do is test the various parameters. So these are the various parameters. So, global changes in methylation. We think this happens. What happens when we measure it? Yes. The answer is always yes, or else it wouldn't be on slides. If you think about it, okay. On the way down, they turn off their DNA. On the way up, they turn it back on. You can sequence all the DNA with Illumina sequencing, or you can look gene by gene. We've done both. This is true of this animal, the ground squirrel. It's true of all the animals that we've measured. What about histone modifications? In theory, histones can be modified and you can turn things off. Does this actually happen? Why, yes. This is an old paper which I love. We've probably published 50 papers on this phenomena since then, but this one's my favorite because we didn't know it was going to happen. Histones get supercharged. They grab up DNA, tie it up, and you can actually see the DNA shrinking. So warm DNA is all active and working around, and then new DNA. Do any of you remember Drosophila salivary gland experiments? It was when DNA was first thing. It's crazy. You can see this physically written on the cell. All these enzymes can be measured, all the modifications can be measured. You separate out proteins by two-dimensional gels and then sequence them. It's just business. And there's a fingerprint for this. We don't have to measure every histone and every modification in every animal. There's a fingerprint. Do phosphohistones go up or down? Does histone deacetylase change up or down? Do acetylated histones increase or decrease? Is there more or less HDAC activity? And how's RNA polymerase doing? These are fingerprints that everyone can measure, and you can't escape this. If all of these things are happening, you may already be, not a redneck in this case, but you may already have your genes turned down. This is what happens. It's so cool, it works on everything. And the third one, which is our current favorite, is microRNAs. MicroRNAs are molecules you have in your genome. You only turn them on under specific conditions, and they reach out and turn other things off. It's absolutely Mother Nature going, what have I got to turn off all the genes again? Hey, haul out the microRNAs. It's another use of a molecular tool. So microRNAs are used for other things, but when they went for metabolic depression, this was perfect. You already had this in your toolkit. You have microRNAs, you have enzymes that make microRNAs, you have microRNA levels you can measure, turning it all off. It's an amazing phenomena, wonderful to work on. You have to know some bioinformatics, or more likely my students have to know bioinformatics. And the first question is, okay, microRNA is a good idea, there's that sperm thing, I'll give you that, but really, every tissue and every organ? Why, yes. MicroRNAs have multiple names, there's 750 of them, and what happened really early on is we measured them all, and then we looked at their fold change, and then we looked at their effect, and then we looked at what happens in our animals. These are the easiest ones to push your head around. This is a microRNA that goes up significantly. It stops making new muscle cells. Well, that makes sense. You're hibernating. You don't want to make new muscle cells. You really don't want to make new muscle cells. Didn't you hear me the first 2 times I turned off myogenesis? So, many microRNAs will attack one pathway, and many pathways are attacked by one microRNA. There's some microRNAs that are just off, damn it. The cell cycle is turned off. We dug into the cell cycle, and it is. There's hundreds of proteins in a cell cycle, and you know how complicated it is. They reach in with microRNA, shut it down, and then when they warm up, they just get rid of the microRNAs, and everything's fine. Hypoxia sensitivity, making new red blood cells. So, as we've gone deeper and deeper into microRNA, there's more and more. Again, my favorite slide. You have a cell cycle. If I reach in and stop your cell cycle, your cells get dead. You have to run them. If they don't get dead, they'll get cancer. My animals stop their cell cycle, 2, 3, 4, and then start them again without damage. It's a cute trick. We don't... Okay, here's the other thing you do. If you write a grant with some questions in it, and you answer all those questions, they're not going to give you another grant. If you write questions in, and then your next grant has even cooler questions, they give you more money. So you can imagine at my age, I've done a lot of that, baby. You want to know how to survive in the mafia? You listen to the old mafia guys, not the pumped up, sleek-haired boys outside the Ravenite Social Club. This is a trick. Turning off your cell cycle without damage is a trick. Here's what they don't do. If you look on the cell cycle, they don't do G0 escape. They don't do something stupid. They actually are able to regulate hundreds of proteins in a way that we didn't know about before. And turning off your cell cycle is important in organ preservation. If you take an animal and just simply dehydrate it, the microRNA levels change, and there's a differential expression, and you can shut off metabolism just by dehydration. One of the best ways to preserve human organs, and one of the hardest to control, you take out the organ and you dry it down just a little bit. Drying it down drops your metabolism automatically, and then with this dried down heart, it actually lives longer, but it's really hard to transplant. The kidney people started this, and the heart people are doing it now. The reason is when you dry down a human organ, the microRNAs don't change differentially. All they do is increase in concentration because you have less water. And so this is an animal that dries down and can reorganize its metabolism. What about the enzymes that make microRNA? Those enzymes can be modified. It's like any other pathway. Dicer and Drosha, Argo1, Argo2, it doesn't matter what their names are. You can get tissue-specific modifications which change the microRNA. So in an animal that freezes, every organ is on its own. Its brain does a different thing, and that will be optimal for cryopreserving brain, heart, lung, liver, skin. Lots of people want to cryopreserve skin because with burn victims and things, you have to have skin. You have to have skin ready. You can't really skin an intern. You have to have frozen skin in tubes, so we work a lot on skin and see what prepares it. The reason I've left this to last, even though it's your favorite animal, is that microRNAs in you and all vertebrates are the same. If I do an array on you and a cow, you're the same. You've got the same microRNAs, the same number and amount. When you go to invertebrates, they have totally different families of microRNAs. We don't know why that is, but in the transition that what Lancelot's made, you just changed over everything you used to do with microRNA and now do something different. We were interested in that transition, but in reality, we had to develop new techniques to do invertebrates. So if you do invertebrates and want to do microRNA, you're going to have to be a little more clever. But this is how you do it. What about the weird things that marine animals do? If you take a littorine, a simple littorine, they can freeze solidly and survive from our northern climates. They can go anoxic and survive. And if you drill a tiny hole in their shell, they can actually dehydrate wildly and survive. They turn on a set of microRNAs, and these microRNA functions are the same as invertebrates, but their sequences are different. So invertebrates have a microRNA of a different sequence. When you look at the vertebrate equivalent, they do the same thing with a different sequence. It's going to be fascinating for new people to look and see how that happens and how different microRNAs can have the same function. You want to promote maintenance. You want to promote antioxidant metabolism. You want to not have your cells die, and you want to promote anti-apoptotic mechanisms. So the microRNAs we found in these animals are extremely functional and useful. And as new labs come on board and start doing this on the vast numbers and different types of marine mollusks, I think we're going to know a lot more. My money is that it's going to be the same ones, and even more people will cite me. But if you look at their liver, it's totally different than their muscle. Invertebrates especially, but a lot of vertebrates, your muscle doesn't have the same problems and the same deal as your liver does, and there's tissue-specific changes. We do not know how this occurs because we only know about Dicer and Drosha. We do not know how you turn on different microRNAs and still have the same function. So hidden under this is some are like foot, some are not like foot. And how do you specifically turn on and turn off microRNAs in marine invertebrates? It's a wide open field. You need a PCR machine. Do you have a PCR machine? There's no reason why you're not doing this full time. I have 6. They're in use all the time. This is the magic pathway. Within your genes are microRNAs that are encoded by your own genes. Some people say they're intron-exon overlap. Some people say they're from viruses or whatever. You copy them out when stress occurs, then you polish them. You polish them, change them, cut them down until they're perfect. Fewer microRNAs are made, but if you look over a time course of stress, you can see this one, and you can see this one, and you can see this one, and you can see the transcription factors binding to DNA that turn on this one. You can measure Drosha and Dicer, and that's useful in itself. If you have a stress and you're asking yourself, are microRNAs available? Boom, do a Western blot. If these guys are up, you're on the right pathway, and you don't have to do a million experiments. But your microRNA comes out of the nucleus, and it's in a really bad neighborhood. You either join this gang or you join that gang, because the half-life of microRNA with the nucleases in the cytoplasm is almost none. It's nice to think that young people in difficult areas could go to school and learn. No, you got to have a gang to protect you. This gang grabs up message RNA and cuts it. You've made message RNA, it doesn't care, because the cost is to make your message into protein, so they cut it up. This actually confuses the message RNA. These message RNAs are found in something called stress granules. So if you look at your cell that's under stress and you find a protein prison, this is just a prison, it's made of proteins, TIA, TIAA, and you find it full of messages looking out going, free me, then you've got stress. These prisons form during stress, and as soon as a ground squirrel wakes up, as soon as a frog melts, as soon as you give oxygen back to a snail, boom, they escape the prison and they're still perfectly there. Your cells actually have them. There was a 1979 paper that showed what happens when you starve mammalian cells. It's what gave us the original idea. You did it was just brilliant and nobody followed it up. Dicer, you could measure this. This takes 2 hours. What about other marine animals? They do amazing things. There's too much salt, too little salt. There's too much poison in the water, not poison enough. Rain falls and garbage comes in. Yeah, microRNAs can be a thumbprint of everything. This is a sea cucumber. The reason that people work on sea cucumbers is they're very, very popular in China. So we have grants in China because they eat these things. You've never eaten one, they taste like seaweed-flavored bubble gum. They're god-awful, but they grow them. They grow them in huge fields in the water, and it's getting too damn hot. So why aren't they reproducing when it's hot? They actually go into estivation. So they're a ground squirrel in the warm. And when we look at a given tissue like the intestine, or when we look at the brain, what happens is they're doing what a ground squirrel does in the cold. We've done proteomics that'll come out. We've done phosphoproteomics that has come out. We've done message RNA. MicroRNAs are really critical, and they're not joking. They turn on a set of microRNAs to turn on their metabolism that would stop a horse in mid-gallop. This is the biggest change you're going to see, and we can't reverse it. So when you take these animals and try to reverse it, they're shut down at temperatures. They're going to have to find a new way of growing these animals, and they're very popular. Oh! This is a paper to which Brad referred. So instead of doing a nice simple project, which is a couple of weeks and then a paper in a high-quality journal, you get on a plane, a little tiny plane, and fly in high winds from Phoenix to Guaymas, Mexico. Do you know why you do that? Because the NSF wouldn't allow me to drive from Phoenix to Guaymas, even though I hate airplanes, because people would kidnap me on the road, and the NSF won't pay money if you drive through Sonora. So that's a thing. Brad's fine. He goes to San Diego, he drinks lattes, and then he takes a big plane. This is a squid that's on the surface. This is a squid that's on the bottom. This is a squid, and I should have known this, it's called the red devil killer squid. Come on,